All right, so I think we're going to go ahead and get started. Let's take a minute to breathe. All of the speakers, make sure that you are ready to go. And we're going to go ahead and get started here today. So once again, welcome everyone to the 2021 Summer Research Symposium. And we'd like to get started off with our first speaker of the day. Courtney Ledford, are you here with us today?
I am, thank you. Let me get my computer set up and I'll start sharing.
Awesome, wonderful. Yeah, I'll let you go ahead and start sharing. That's perfect. All right. Thank you, Courtney. Go ahead and get started.
So I'm presenting my project, which is looking at the effects of Apremilast on binge drinking and circadian gene expression in mice. And I am working with the Osborne lab up at the Veterans Hospital. So a little bit of background about circadian processes. So circadian processes and the molecular clock found all throughout the body, and they control different functions such as hormone regulation, immune system, metabolism. And for my project, I am looking at the VTA, which is a brain region responsible for addiction. And then to connect this to alcohol, individuals with alcohol abuse are more likely to have disrupted circadian rhythms, and then disrupted circadian rhythms leads to an increased likelihood of addiction and relapse. So for this experiment, the goal is to identify a treatment that can alter the molecular clock to help people with alcohol use disorder. And so this experiment is using Apremilast, which is a PDE4 inhibitor. And the goal is to reduce binge drinking and high drinking the dark mice. And these mice are bred to drink until intoxication. And the goal with this is to decrease the binge drinking. The second question is to increase levels of circadian gene expression in the VTA of aged-in mice with a history of chronic binge drinking. And the goal of this is to increase circadian gene expression in the VTA. Oh, right. Sorry, just a moment, I'm a little nervous. And so for this experiment, we're testing Apremilast, which uses a 8-week drinking in the dark method, where we use both male and female high drinking in the dark mice. For days 1 through 3, they were given 2 hours of ethanol access. On day 4, they were given 4 hours of ethanol access. And days 5 through 7, they were only given water access. And so for the 8 weeks, weeks 1 through 6, they were only given baseline. So we're just collecting baseline of their average ethanol intake. And on week 7 and 8, they received the treatment of either Apremilast 40 milligrams per kilogram or the equivalent in a saline injection. And the tissue was collected at 2 different time points, which is Zeitgeber time 3 and Zeitgeber time 15, which is to just account for the different peaks and troughs of circadian gene expression. And the factors for analysis we're looking at sex, treatment, and time of day for this experiment. And for the methods being used, we first did the drinking in the dark 8 weeks, then we collected the brains, then we took the VTA tissue punches, the mRNA was then isolated, the cDNA was then synthesized, and then right now we are looking to measure the clock gene expression and then move on to data analysis, hopefully in a week or 2. So for expected results, we're hoping that Apremilast will increase circadian gene expression in the VTA, which will then ameliorate the effects of chronic binge drinking on the molecular clock. And this is important because the molecular clock regulates expressions of thousands of other genes, which are often disrupted by alcohol use. And so the Osborne lab saw that Apremilast reduced chronic binge drinking in the VTA of high drinking in the dark mice. And so for this experiment, we're seeing if there's an increase in circadian gene expression, because we already know that Apremilast shows a decrease in drinking. And so for future directions, we're hoping to compare gene expression from the VTA to other brain regions important for rhythms, such as the suprachiasmatic nucleus and the nucleus accumbens. And I'd like to thank the Osborne lab for making this work possible, as well as the Build Exito program, the McNair program, and all of you for being here this morning. Thank you. And then if there's any questions.
Wonderful, thank you. That was a great presentation, Courtney. Yeah, let's give Courtney some virtual claps. Awesome. Yeah, really great job. Really nice presentation. Yeah, and I'd like to open it up if anyone has any questions for Courtney about her presentation. And if you have questions, go ahead and put those in the chat. I can start us off with one question. I may have missed this, but with the drug Apremilast, I was wondering if it has an effect on reducing the intake of any other substances. You may have mentioned this already, other than alcohol intake.
The Osborne lab is actually looking at nicotine. They've also looked at cocaine in the past. So we are looking at different drugs of abuse. Apremilast is currently approved for different inflammatory diseases.
Awesome. That's really interesting. It's also interesting to hear that it's already been approved for other diseases. Does that make it easier when you're looking for candidates for drug targets for your work?
And we are working with other labs, and Apremilast is in human trials as well for drinking.
Great, great.
Thank you.
Yeah, any other questions from the audience? Really, really interesting work and really important work as well. Yeah, we have time for maybe another question if anyone would like to ask one. I'm so interested in the topic. I was wondering if you know if there have been other drugs that you've worked on in the lab, or if any others have tested other types of drugs in this field.
Yes, we've looked at different drugs. This specific drug is targeting the circadian processes, but we have tried other different things. Sorry, and I see a question in the chat, so I'll answer this question. Is there any effect on water intake? So this was a beginning question, and we didn't want to do too many mice yet because we're working with mice. So if everything goes according to plan, eventually we might get a water group, but we did not have a water group for this experiment.
Awesome, thank you. I'm seeing another question in the chat. We might have another little bit of room here. There's a couple questions. Is there different species of mice that have been used in your work?
There are, and the Osborne lab works a lot with these high drinking and dark mice, but we also do some other different mice that are not bred to drink until intoxication, but these mice are bred for chronic binge drinking.
Oh, that's really interesting. And then one last question that I'm seeing in the chat is, have there been any unanticipated health benefits to the mice?
For the mice, not that we've seen yet, but for a different medication, Rolapam, which is also PDE4 inhibitor, in humans, it has effects such as nausea. So that's why we're looking at Apremilast. So far, no effects in mice.
Awesome, great work. Really, really interesting, and just so, again, so important. Thank you so much for your presentation. Let's give Courtney another round of virtual applause. Thank you so much. All right. Well, we will give just a minute or so for our next speaker to get set up. Our next speaker is going to be Nancy MacKenzie, and I'll give you just a couple seconds to get ready. Let me know when you are feeling ready to go.
So, hi everyone, thanks for coming. My name is Nancy MacKenzie, and I'll be presenting my research out of Christoph Tuscher's lab today. And my topic is evolving reservoir topology using the genetic algorithm HyperNEAT. So, what is our goal? What are we trying to accomplish with this work? A typical neural network is static, and the amount of neurons have to be chosen from the beginning, and the quantity is a bit of a guessing game. We want to evolve neural networks that reflect the task at hand. By studying the resulting architecture, this will tell us how neural networks grow to solve problems. So distilling this, we want to grow neural architectures that gradually become more complex and are biologically inspired and neurologically plausible. To achieve it, we're using reservoir computing, which is a recent approach to machine learning, and the HyperNEAT algorithm, which is a state-of-the-art genetic algorithm inspired by evolutionary and genetic processes. So the hypothesis is, by gradually increasing the size and complexity of the network to the extent that the task requires, we'll be able to build more optimally performing efficient networks that exhibit modularity that's reflective of the task. So what is a reservoir? A reservoir is a type of echo state network. Echo state networks evolved out of recurrent neural networks. Recurrent neural networks have feedback loops. So this allows the internal nodes that make up the reservoir to possess memory or state and process sequential tasks. Echo state networks can be adapted to a wide range of tasks, such as speech recognition, motor learning, natural language processing, working memory, etc. They're also used to process EEG data. So a traditional echo state network has fixed random connections between nodes in its reservoir. And these output weights over here are the only thing that's trained in an echo state network. In typical neural networks, you're going to be training all of the weights. So an echo state network has lower training complexity. More recently, researchers are finding that creating subreservoirs or modularity increases performance. However, finding the optimal modularity and size for that desired performance proves to be a challenge. Okay, everyone take a deep breath and let's say the full name. Deep HyperNEAT stands for Deep Hypercube-Based Neuroevolution of Augmenting Topologies. It's a genetic algorithm taking its inspiration from evolutionary strategies to evolve how artificial neural network structures grow using genetic operators and indirect encoding to achieve the highest fitness for the task at hand. Genetic operators are at the core of genetic algorithms. We have selection of the fittest individuals, in this case, best performing neural network structures, crossover, which would be recombination of parent genomes to form a new offspring, and mutation. These figures on the left are the phenotypic outcomes of 3 of the mutation operators in deep HyperNEAT. We have incrementing depth is adding layers of neurons to the neural network. And then we have incrementing breadth, which allows for parallel processing. And then we have adding new connectivity, which allows for more neurons to be connected together. So what's been accomplished so far? So far, I've used Python to build and test an echo state network. So in this enlarged figure on the left, what we have is the reservoir with no inputs. So the reservoir starts out very chaotic, and then once the perturbations of the reservoir die down, we put in inputs, and that's what you're seeing in this image on the right here. So each vertical line represents an input being sent into the reservoir. The input is then clamped or held there until the perturbations die down and we can average the output from the reservoir. I've also learned a lot about deep HyperNEAT and the algorithms leading up to it. NEAT actually started in 2002, and since then quite a bit has been added, including indirect encoding and the ability to grow larger substrates. So what are the next steps? Well, one, we want to establish that the evolved reservoir is working. This will be done by testing the deep HyperNEAT evolved reservoir with the classification test present in the library. And once I've established that it's working, we'll use deep HyperNEAT evolved reservoir. See, it's a lot of words, I know. To solve more complex tasks. An example would be replicating canonical neuroscience tasks or solving multiple tasks in different orders. Which leads to the next step, evaluating the resulting structure of the reservoir. Do biologically realistic connection patterns emerge? Does modularity emerge, and how does it vary according to different tasks? How does previously formed modularity inform future tasks? And perhaps we can compare and contrast our results with MRI connectomics data. So at this point, I'm ready to take questions, and thank you again for listening in.
Thank you so much, Nancy. That was an amazing presentation. Really great work. Yeah, definitely over my head with a lot of the words that you described. So yeah, really awesome. It sounds like there's a lot of work that you've been able to accomplish over the summer, and just a lot of really in-depth and complex, kind of ideas going on there. So this is really, really nice work. Yeah, I'd like to open it up to the audience to see if the audience has any questions they'd like to ask about Nancy's presentation.
What was your most challenging part of this experiment?
I think a lot of this has been quite challenging. I just started learning Python last summer, and there's also been quite a lot of new math notation to learn. And I had an sort of outside interest in machine learning and neural networks, but it's a whole new thing to step into it. And it's been so cool because I've really learned a lot about how you can use different math functions to like create separability between classifications. And this is also just super interesting because it falls within computational neuroscience, which is something I'd like to get good enough at computing to get into.
Awesome. Wow. This is really, really interesting, Nancy. I see one question in the chat, and this is a good question for all of us that aren't used to this, is what is Python? Can you explain that a bit?
Python is a programming language, and it's really user-friendly, I think. And it's a programming language that's really kind of the rising star in STEM right now. Thank you for your question.
Great, yeah. And I'll ask one more question. I think we have a little bit more time, but you kind of touched on this with your next steps. But could you elaborate just a little bit more about what kind of the big picture goal is in using the deep HyperNEAT? I might be saying that wrong, but
deep HyperNEAT's right. Yeah. So the big picture goal would be to be able to look at how function and form are interrelated, and kind of using these tools alongside neuroscience data that we have to really increase that crossover between neuroscience and computer science. I think that there's so much data and so much information with the brain that like having this these extra tools are going to be vital, because there's a lot of data to process, and we're going to be able to use machines that are better at seeing patterns, and then really combine human expertise with these machines in figuring out more about the brain's architecture.
Wow, that's amazing. I see one more question in the chat, and that is, what does it mean for a neural network to be biologically plausible or neurologically plausible? If you can answer that one.
I think that that's also something that we're all still figuring out. Some of the things that come to mind are reservoirs are supposed to be a little bit more plausible than, say, there's something called the feed-forward neural network, where there's no information can only go left to right. So there's no information going back the other way, which isn't very brain-like. And we can have feedback loops with recurrent neural networks. And so we want neural networks that have memory, take in these time-based inputs. But there are also things like what's called activation functions, and how things are summed, kind of like in a neuron, it acts on Hillock. So we really want to look at what we're finding out in neuroscience to inform the science that we're doing with neural networks and machine learning more. But that's just a really, really huge question. So I know I only said a couple things.
Oh, that's amazing.
Thank you for your question.
Thank you so much, Nancy. Let's give Nancy another round of virtual applause. That was a really great presentation.
Thank you, everyone.
Thank you so much for your work. Awesome. We have just a couple minutes before our next speaker. So our next speaker is Thandose Kalinda. I'll give you just a minute or two to go ahead and get prepared and get your screen set up. Let me know when you're ready. That looks great.
I think I'm ready. Can you guys see that?
That looks beautiful. All right, go ahead, Thandose.
Hi. Okay. Hi, everybody. Thank you for coming today. And my name is Thandose Kalinda. I am in Dr. Borikia's RLC in the ED at OHSU. My topic today is, can clinical decision support tools or CDSs improve provider prescription practices in the ED? What is atrial fibrillation? Atrial fibrillation, or AFib, is one of the most common cardiac arrhythmias, and it occurs when the electrical signaling system of the heart is disrupted, causing the atrium or the upper chambers of the heart here to contract in an unsynchronized manner, slowing down the flow of blood in the heart, allowing it to pool and form clots. The longer the individual is in atrial fibrillation, the higher the risk or the chances that these clots can dislodge and travel to the brain and cause a stroke. These comorbidities are, like in many chronic illnesses, increase the risk of stroke. And these comorbidities here are integrated into a stratified risk assessment scoring tool called CHADS-VASc2. And it determines the patient's risk of stroke. What are clinical decision support tools? These are patient-centered guidelines that will help to provide an ED provider with up-to-date patient information that are integrated also into an electronic system, since everybody uses electronic medical record networks right now. So they provide alerts and notifications when the patient comes in. We also hope that they aim to reduce provider hesitancy when it comes to prescription of anticoagulants in the ED, thereby increasing patients' long-term adherence to anticoagulation medication. That would, in turn, reduce their risk of stroke, and also reduce the patient's risk of hospital costs, increased hospital costs when they come in due to complications of AFib. The objective of this study was to determine where the current gap is in physician prescribing practices at the moment. We hope that when we find this, hope that the study can give us like a baseline that will help us determine whether CDS tools can help improve provider prescription practices in the ED. In the study, we used electronic medical records from 2015 to 2017. And from those, we just decided to figure out which ones were new onset and which ones were recurrent cases of AFib. And from there, we looked at what the type of intervention was provided and whether an anticoagulant was prescribed in the ED. The inclusion criteria included patients that were presenting in the ED with symptoms of AFib and were actually treated in the ED. Exclusion criteria was determined by patients who came in with AFib symptoms, but were not treated in the ED for the AFib, but for another more emergent health problem, such as sepsis or anything else. 280 charts were screened, and from there, 60 were analyzed. We found that there were 11 new onset cases of AFib, and there were 49 recurrent cases. We looked at what kind of intervention was provided. There was chemical cardioversion using these medications, electrical cardioversion. Rate control was to bring the patient's heart rate from above 100 to below 100. And none is when the patient came in with AFib symptoms, but during an evaluation, reverted back to normal heart rate and rhythm by themselves. The frequency, as we can see here, the frequency of prescription of anticoagulants in the ED was 4 for new onset cases and 7 for recurrent cases of AFib. As we move forward, we wanted to see whether with this study, where the gap was and why, as clearly we can see there are very few anticoagulant prescriptions being prescribed to both new onset and recurrent. And we aim to reduce reduce the deferring practice of anticoagulant decisions, because once it's deferred, the provider does not know whether the patient goes on to actually follow up with that provider out of the ED to get the prescription started. Therefore, we needed to determine whether the integration of these tools would help streamline and increase provider prescription practices in the ED, as this sets up the patient to adhere to long-term medication of anticoagulant right from the start, and not wait for another prescriber to start the prescription process. The future plans would compare newer cases compared to the older cases that we've looked at, and to test if the hypothesis to see if the CDS tools can change provider behavior, specifically prescribing practices in the ED, as this is like the ED can be used as a teaching moment for both providers and for patients when they come in, and to discuss and make sure that everybody's on and determine patient and prescriber satisfaction over the next 5 years using the same studies, using the CDS tools once implemented early on in the ED. I would like to thank Bill Exeter and NIH for providing this opportunity to me and everybody else in the Bill Exeter group, and to Dr. Borikia for taking me under her wing. It's been wonderful learning from her. It's always a learning moment. And to Joy and Nancy and our team in the office, they're always there to answer questions. And I thank you so much for that. And also to everybody in the Center for Policy and Research and Emergency Medicine at OHSU. Thank you.
Awesome. Great work, Thandose. Beautiful. Yeah, great presentation. Really, really interesting work. And again, really important. Yeah, I can see a lot of benefits to the community and to just kind of the future of healthcare in general around atrial fibrillation. So really, really beautiful work. Yeah, I'd love to open this up to questions from the audience if you have any for Thandose. Oh, I'm seeing some questions. Yes, the first question I see here is, how does this interact with current best practices around TIAs and strokes?
Well, this specific study was looking at to find the gaps of in prescription practices for AFib patients when they came in. The TI and MIs actually increase the risk for patients with, especially if they have AFib and they come in with these other comorbidities, it actually increases the risk of stroke. So we hope to determine, like, well, if we introduce these tools, can they also reduce the risk of, patient getting an MI, further on in the future?
Awesome, awesome. Another question coming in is, what are you guys doing currently in this study, and what step are you on?
This is a retrospective study. It's an ongoing longitudinal study, and my part was to find out what within this, the certain years, which of these patients are once again coming in with AFib patients, how have they been treated, whether they've been prescribed by whether they've been prescribed an anticoagulant while they're in the ED prior to discharge. It is an ongoing study, so I'm sure someone else will step in behind me and pick it up from there and ask another question to build up upon this. So that's a good question. It's an ongoing, and we look forward to see what else will come up from this.
Great, great. We have just a couple more minutes. Another question is, and I might mess this up, is metoprolol and aspirin used in conjunction?
Well, aspirin is what's actually not, sorry, I always mess up. It's not really accepted by, pardon me, Dr. Kia, correct me if I'm wrong.
Great.
Yeah, it's not currently a accepted regulated anticoagulant, is that correct?
It's not part of guideline recommended therapy. It is an anti-platelet agent, so it's a little bit different from the anticoagulants, which include things like rivaroxaban. So it's a different type of medication, but it is not part of guideline recommended therapy It is often used though with metoprolol, which is the rate control medication that brings the heart rate down. So they have different actions on the body. And I also put in the chat the actual clinical decision support tool. It's a link to it that we'll be using and be integrated into the electronic health record. We are currently on step 0, and that's what Thandose has been working on, which is the pre-implementation part of the study and getting baseline data. And then we will start with step 1, which is we have multiple steps where there's education and triggers and lots of other types of aspects we'll be studying as part of the implementation of the tool to see what works best for providers and patients. So what Thandose has done is a key part of doing any studies, figuring out what is the gap in care before we actually embark on doing the actual study.
Wow, very, very interesting. Very nice research, Thandose. We're very, very proud of you. Let's give Thandose another round of applause. And if you still have questions for her, please reach out to her. I'm sure she'd love to answer those outside of this. Let's go ahead and get ready for our next speaker, Jose Camara-Lavadores. If you're ready, go ahead and let me know when you're all set up and ready to go.
Cool. Some pretty great presentations so far. So my name is Jose Camara. I'm here representing the Podravsky lab at Portland State. So we are a developmental biology lab with a focus on embryology. So what that means is that we study eggs, specifically the eggs of these fish called Astrophilus limnaeus, which is a Venezuelan killifish of tropical climate. This here is my depiction of it. And in the background, we see some of the work that's been going on, which can range from biochemistry to gene expression, as well as physiological changes. So the reason, there we go. So the reason why we study this fish and the reason why this fish is so dang special is because it's capable of doing this thing called embryonic diapause, where here it says it's a state of metabolic arrest, but really it's more of a state of metabolic slowing, for lack of a better word. During this stage, the cell does not do any active growing. There's low amount of energy being spent, and there's no protein synthesis. This, of course, is an evolutionary adaptation that allows survival in harsh environments. One of them being drought, which is depicted here. Here we have the dry season, where you're not really going to see any fish swimming around, in the rainy season. So they pop up periodically when the rain and all the conditions are favorable. Down here we have Astrophilus limnaeus in all its glory. This is a male. A lot of coloration, beautiful fish, happy. So this leads us to the question, in my personal interest, is could host microbial interactions support this unique physiology of diapause? So when I sat down with Dr. Jason Podravsky, when we met for the first time, we put together this project where we would characterize the bacteria that are living inside these fish and also the embryos, where my idea was kind of just plating them and seeing what grows. But then later on, we would talk to Ann Thompson, who steered us in the right direction and said, yeah, you should do DNA extractions, sequence that, and PCR that. So with this, we decided on doing sterile technique for dissections, DNA extractions as well. Later on, we're not really quite here yet, but we're getting there. We're going to do qPCR, where we amplify the microbial DNA, and then we're going to sequence this DNA to see what kind of microbial species are present. So we're seeing what we find. This is the journey, and this is what I've been doing in the lab. It's been a lot of dissections, and like I said, under sterile conditions. So here in the center, we see a dissected female, where she has the ovaries, as well as the swim bladder exposed. Here we have the ovaries blown up a bit more. You could see, so we were really expecting to see more developed embryos down here at the bottom, closer to the cloaca, but you could see that it's really not the case. They're kind of scattered throughout. You see more developed embryos here, underdeveloped here. This is the adult testis, which would take the place of the ovaries here if this were a male. And these fish are tiny too. And this is, I like this picture because it kind of shows the extent of the quality of work that we've been able to do. This is a swim bladder. So it's like a very thin membrane balloon of gas, and it's unruptured, undisturbed, and you can see it here. Pretty cool. We were working under a biosafety cabinet, which you could see. This is homogenized tissue. So when we collect these tissue, we extract them, we homogenize them, meaning we use both physical and chemical ways of disrupting these cells. So when we measured our quantity and quality of DNA, we used something called a TCAN, which takes your DNA concentration, and it also gives you light absorbance ratio, which will tell you the quality of it. Here we used a DNEZ blood and tissue kit, where you could see that the DNA concentration is larger because it includes both the DNA of the fish as well as microbes, where this one here is a DNA microbiome kit, and you see a very drastic decrease in DNA concentrations here. This is because this one gets rid of the the fish DNA. So kind of where we're at this point is we're getting ready to do PCR, which will be a lot of fun. We just got some primers. It'll be fun to play with that. And yeah, so I'd like to thank the Podrabsky Lab, Dr. Ian Thompson, Dr. Nacho de la Higuera from the Stedman Lab, Dr. Dan Mills, my career mentor, as well as Bill Dexito, and all of you for taking the time to listen to the things in life that bring me joy. Thank you.
Thank you so much, Jose. Yeah, that was really great research, and I love your artwork at the beginning. That was really awesome. Yeah, really, really nice work. Yeah, I'd like to open it up to the audience first, if there are any questions about Jose's research. I have a question I might be able to start us off with, and might be a little bit more technical, but kind of going into your DNA extraction, I was just wondering if you've seen differences in the amount and quality of DNA that you've been able to extract from different tissue types from the different parts of the fish.
We're definitely seeing a lot more DNA in the embryos than we are seeing in actual tissue, but definitely you see a good amount of yield in the what is it called? The gonads, so the ovaries and the testis.
Oh, that's really interesting. I'm seeing in the chat that there is a question. If you can explain, what is PCR?
It's a polymerase chain reaction where you're taking a segment of DNA and you're amplifying it, so you're making copies of it. And, it just makes it so that you could see that it's there. That was a kind of a bad explanation, but it is there.
Awesome, awesome. And then another question I'm seeing in the chat is, do you know what you'll be using to perform the DNA sequencing?
Not quite yet. I think we're looking at I think it's called a nano drop from Oxford. I think we will be purchasing that for this experiment. But I think that's probably what we're going to do. Otherwise, we'd probably end up sending it to somebody to sequence. But ideally, we'd be sequencing it ourselves.
Awesome, awesome, great. Yeah, and I'm seeing, yeah, maybe someone was recommending maybe nanopore. And we have a couple more minutes. The only last question that I had, and I'm just curious, is you talked about the difference between the whole DNA extraction and then kind of looking at the microbial DNA that you were extracting. I was wondering if you or your lab has looked into the different types of microbes that you've been able to pull out and if those differ.
We haven't done the sequencing, so we can't say for sure what's going to be there.
Yeah.
And it's a little hard to say too, especially with bacteria being able to share certain genes and send them off to each other. So for those types of genes, it'll be a little bit harder to characterize, but definitely sequencing will help us determine what's going on there.
Awesome. Well, really nice work. And sounds like you're learning a lot of really great techniques that are going to be useful in this project. So yeah, great job. Thank you so much, Jose. Let's give Jose another round of virtual applause. Really awesome work. Thank you so much for your presentation. All right, and we have one more minute until our next presenter is ready. And our next presenter is Tyler Robin. If you are here and would like to get set up, and you can go ahead and start sharing your screen and let me know when you're ready to go.
Okay, I'm unmuted. Perfect.
Awesome.
Okay, thank you so much. Good morning, everyone. Thank you all for being here. My name is Tyler, and my project is comparing heavy metal content found in spinach grown on the roof and ground sites at Portland State University. So just as a quick introduction, a green roof is a roof with purposeful living vegetation. There are many benefits to green roofs. Just a few benefits could be reducing noise pollution, increasing roof lifespan, reducing air pollution, utilizing previously unused space. Also in Portland, there's a Portland project where if you're within a certain height and within a certain distance of the downtown area, you have to have some sort of green roof on your building. So that's another reason why it's important to talk about green roofs in this area. So my question was, well, what about growing produce on the roof? And there are definitely some benefits of doing that. That would eliminate contamination from shipping produce from wherever to urban areas. It could potentially eliminate pollution from shipping. And it could potentially lower prices and reduce prices and offer a cleaner vegetable for urban areas where there's lots of people. There are definitely some concerns about growing produce on the roof. We don't fully understand how pollution affects leafy greens grown on roof and roof levels. We have to keep in mind that they can be heavy, and there potentially need to have shallower root systems when growing on the roof. So my research question was, how does growing food on the roof versus ground compare when it comes to heavy metal contamination? So for my project, my design, we used spinach beds that you could see here. And they were cat trays that were bought at the dollar store with potting soil inside, and we planted them in a 3x7 planting pattern with a temperature sensor in the middle. We watered them weekly unless it rained with the same water to limit variables. We also did air quality tests weekly and weekly monitoring with growth pictures and measuring of the actual leaves as they grew. We used 5 rooftop locations and 5 ground locations within close proximity to each other at varying heights. And this is a map of the ground and roof locations. For my actual methods of the extraction, we separated the leaves from the roots, and then we dried and weighed both the leaves and the roots. And then we did the actual extraction where we ground up the leaves into a powder. We digested the leaves in a hydrogen peroxide and nitric acid mix as we heated them and shook them. And then we used an ASV and an ICP OES machine to determine the concentration of metal found in those solutions. So for the actual growth results, the cotyledon growth is the pre-leaves. It's a good indication of nutritional value in the early stages of the plants. And you can see that week 3, you definitely see a spike in higher cotyledon growth on the roof versus the ground. More growth results. We also saw the same kind of thing with the spinach leaves on the roof versus the ground within week 5 when they actually started to grow a little bit more. For the mass results, keep in mind, we actually only had 4 roofs and 3 ground locations produce spinach that we were able to weigh and do the extraction process with. So, but those averages, we did see that there was a higher yield of both leaves and roots on the roof locations versus the ground. So for the actual heavy metal results, we did see higher amounts of cadmium, chromium, and lead in the ground locations versus the roof. So in conclusion, overall, the roof locations grew more spinach and had fewer heavy metals. The ground locations did have issues with outside influences, and that could reflect what it would be like growing produce in an urban area with higher populations. There's definitely more potential to be disrupted by people or animals, crows or squirrels or dogs, things like that. And we would need to take that into consideration if we did want to do some urban agriculture. So for our future directions, we want to replicate the study with more ground locations with more barriers in place if possible. Potentially do the study with different plants that grow better in this area, including like kale or chard. The spinach did have difficulties with the heat wave that we saw. So we did lose a few plants for that. Also, I had the idea of doing a study using hydroponics instead of soils, because that would eliminate anything that has to do with the soil, and then we could really compare and see if is it the water, is it the air, is it the plant itself. So here's my works cited. I just want to acknowledge the Build Exito program. I want to acknowledge the Dr. Alyssa Starry's ecology lab, Jesse, who was a previous Exito student who I modeled my project on. He did the same kind of project a few years ago. Alondra, who helped me with the whole project, and the McNair program. All right, any questions?
Awesome. Thank you so much, Tyler. That was a great presentation. I'm really excited about Green Roof. So I was super excited when I saw your title. This is something I've been interested in for a while. So really, really beautiful work. I'm seeing some questions coming in for you in the chat. The first question I'm seeing here is, how does the lead get into the roof plants? Is this from the rain?
So we are not completely sure. So some of our hypothesis is that it comes from air pollution in the area, some from the soil potentially. We really don't know at this point. It's an issue. I mean, so leafy greens they absorb things and they hold metals. So that's why we use spinach originally, is because they're big absorbers of metals. So it really could have come from anything, and we just don't know at the moment.
Awesome. Another question, and this is really interesting, is did the building heights make a difference?
Yeah, the highest yielded spinach bed that we had was the tallest building. But also they weren't within, I still need to do the statistics on this, so I can't say if it was statistically different between the different levels of roofs yet, but we're working on that and it would be really interesting to see. So
yeah,
maybe.
Yeah, that's a really interesting question. Yeah. Another question I'm seeing here is, how did the roof spinach metal levels compare to store-bought spinach?
We didn't actually test that, but we are going to try and do that with our next round of spinach beds. I really want to do, I want to compare potentially if we can't get our ground sites to actually produce, because there was difficulty with that in the previous study as well. We were thinking about comparing the roof versus either store-bought and farmer's market spinach to see if there's a difference between like what we would be ingesting versus like what we would be growing on the roof. So that's another future directions. We have a lot that we want to do.
Awesome. And I think we have one more minute. I'll ask you one last question. Was there higher yield on the roof due to the increased exposure, or were ground and roof location pairs chosen with similar sun exposure? And great job.
That's a good question. So we just had a really difficult time with the ground locations. There are a lot of locations that are more shady. So we tried to pair them as close as we could, but you know, things happened. We were in a downtown area. So, you know, and also we did see different temperatures on the roof. Green roofs are known to be hotter than on the ground. So that could be a difference. Yeah, I see that in the chat. That could have affected the growth. But again, we want to do the study again and replicate it again and try and answer some of these questions. Great
Awesome.
questions. Thanks, everyone.
Thank you so much, Tyler. Let's give Tyler another
Thank
round of
you.
applause for a really great presentation. There's a lot more questions, so please reach out to Tyler if you'd like to learn more about. We will move on to our next presenter. Our next presenter is Katherine Ouda. Katherine, if you're ready, go ahead and start sharing your screen. Let me know when you're ready to go.
So hello everyone, my name is Katherine Ouda, and for the past year I've been working at Dr. Luis Bertassoni's dental research lab on the optimization of a tooth-on-a-chip device, and I'm very excited to share what I learned with you all. To start off, let's explain what's an organ-on-a-chip device. It's a microfluidic platform to study cells in 3D in a more physiological manner. The team at Dr. Luis Bertassoni tried to mimic this idea and implement it into teeth by creating a tooth-on-a-chip device that is also a microfluidic device that mimics the interactions of dental pulp with dental materials and dentin. It also tracks cell response in real time. The tooth-on-a-chip device was fabricated for many reasons, which include that for current cell culture systems, many cells and dental materials are being used, which come with high wasted costs. Also, there's an ethical problem of using animals in humans research. And as we all know, animals and humans are similar, but they're not identical, which doesn't give us very accurate data. And for the first time, researchers will be able to visualize the interactions of dental pulp and dental materials in real time. To commercialize the chips, they can't leak, so we had to optimize the best method of fabrication and seal the chips without losing the biological properties of the dentin. So here on the right, there's an image of a leaked chip. And this shouldn't happen in a real experiment. And we tried to solve this problem. We started by preparing dentin fragments from human third molars. We used an automated saw machine to cut them into dentin discs and used another slow automated saw machine to cut them into dentin fragments. Then we sanded them by hand to get to a certain parameters, like a width of 4.6 millimeters, width of 1 millimeter, 0.6 millimeter, and then the depth of 1 millimeter. Then we had to assemble the devices by using polydimethylsiloxane, PDMS, which is a silicon-based polymer. We used many conditions for the leakage test, and we tried the plasma bonding it either against the glass slide or another PDMS. Then we filled one side of the chip with water and the other with water and food dye with a dentin piece in between. Then we sealed it with Kapton tape and placed it under the microscope for 8 hours to see if any leakage will happen over that period of time. And here are the set of conditions that we used to see what's the best method of fabrication. The PDMS and the glass slide was the baseline, and the others were the different conditions. And here's an image of a microscopic chip, the baseline, just the PDMS against the glass slide. Here's the one with the glue, and here's one with resin. And then here's one just with tape. And here's the last one with just PDMS and PDMS, or half and half. And we can see how the chips with the glue and the resin, they're very messy in comparing to the others. So we had to examine the assembly success rate and the leakage success rate. We assembled 4 chips for each condition, and the success rate for the assembly test tested if the chips didn't stick at all or they leaked right away. And we noticed that the PDMS against the Kapton tape and the PDMS against the other PDMS had the best success rate, 100% success rate. Only 4 chips were assembled and 4 chips moved on to the next test, the leak test. And on the leak test, which also tested if the chips leaked over the period of 8 hours that was viewed under the microscope, we found out that the PDMS against the glass slide and the resin and the PDMS against the other PDMS had 100% success rate. They didn't leak at all for full 8 hours. So in conclusion, we found out that PDMS and PDMS, or the half and half, had the best results for both the assembly test and the leakage test because they had 100% success rate for both. And they are a good applicant for future experiments because they're not messy as the glue and the resin, and they don't have materials like the glue and resin that may interact with future tested cells in a future experiment. So we did another test with rhodamine for the half and half experiment. Rhodamine is a group of related dyes for half an hour, and it also succeeded. It didn't leak at all. It's this image on the top right. And we can see how one side is fully red bright and the other is dark. I would like to thank the Build Exeter team that was funded by NIH. It provided me with this great opportunity, and also special thanks to my RLC leaders and PI, Dr. Christiane Franza, Dr. Louise Bertassoni, Anthony, and all the Bertassoni's labs team. And thank you all for listening to my presentation. Now we're ready for questions.
Awesome. Thank you so much, Katherine. That was a really, really interesting presentation. It looks like a lot of work was done in building those chips. Yeah, I'd love to open it up if the audience has any questions for Katherine about the presentation or experimental design. Okay, I'm seeing one come in. What is the clinical use for the dental chips?
So in the future, as I mentioned also earlier, that right now there's no method that allows researchers to view what's actually happening inside of the teeth. So for example, like for when they do root canals, they're trying in the future to create live cells, live dental pulp cells by using this device and experimenting in one side that will have the dental materials. I'm going to go back to that slide to explain real quick. So for example, like here we have a chip. So one side we can put dental materials and the other side we can put dental pulp, and in between the dentin, and we can see what's happening and track cell response in real time. And we'll see, we can, instead of dental materials, we can put any kind of drugs or anything we would like to test and see how it affects the dental pulp and what helps in the dental pulp regeneration in the future.
Awesome. That's really, really interesting. I do have another question, and this might be a very naive question, but just wondering, how and kind of where do you get the dentin and I guess the teeth that you're getting that dentin from? Is that something that's hard to get enough material to work with for your experiments, or is that something that's been easy to come by?
It's been hard to come by, but it's from people who donate their third molars after they get extracted. And at the, I've been working at the dental research building. It also had, it has some dental clinics. So we get that basically the extracted third molars from humans' teeth that they donated.
Wow, that's really interesting. And it's awesome to think that things that we don't need anymore can be put to such use, you know, in your research that can be used to actually help other people then, kind of full circle. So really great research. Thank you so much. Let's give Katherine another round of applause for that awesome presentation. Yeah, really, really great work. Thank you so much. All right, and we will start moving on to our next presenter. Our next presenter is Evie Thompson. Evie, if you're here and you're ready, if you'd like to start sharing your screen.
Oh, sweet. Alrighty. Hi, everyone. Welcome. My name is Evie Thompson. I am with the Trauma Research Lab at OHSU under Dr. Martin Schreiber. Today, I'll be talking about whether P-Selectin can be used as a marker for platelet activation in trauma patients. So the really big question of all of this is there trauma or is there platelet activation in trauma patients? And can we observe this using an activation-based protein? So why look at this at all? Platelets are one of the key parts of coagulation. So it's kind of important that we want those to work properly. And it's been hypothesized that patients who go through a significant trauma develop acute traumatic coagulopathy. And according to a 2012 study, roughly 25% of trauma patients present some evidence of coagulopathy. And so the chosen activation-based protein was P-Selectin, also called CD62P. And here on the screen, we have a non-activated platelet versus activated platelets. So they really do change shape when they activate. So for P-Selectin, what is it? It's a single chain glycoprotein that is expressed only by activated platelets. And its purpose is to activate other platelets and endothelial cells. And the whole point of this is to form an initial platelet plug during hemostasis. This is when the body is attempting to stop the bleeding going on. The methods that we used were samples were pulled from a previous study that included multiple trauma types as well as injuries. The samples were thawed from -80°C, and those plasma samples were prepared for a Luminex XMAP, also a multi-analyte profiling plate. Some of the demographics from our samples, roughly 75% were male and 25% female. We had a mean age of 53 years old. Injury severity score is a score calculated after the discharge of patients that's based on their complete list of injuries, and our mean ISS was 17. For this presentation, we had 177 samples at baseline, and baseline is admission to hospital, 150 samples for 8 hour, which is the 8 hours after that baseline, 129 samples for 24 hours after baseline, and 97 samples that are in our 48 hour. We also had 3 different trauma types. We had a traumatic brain injury, and roughly 15% of our samples had this kind of injury. We had roughly 63% have a non-traumatic brain injury, but still another type of traumatic injury. And roughly 22% were polytrauma, meaning they had a TBI plus another trauma. So kind of what are we seeing so far? This was the average P-Selectin levels over time across all of our 4 time points. And as you can see, there is sort of this peak at the 8 hour. And this is sort of interesting because it means that we're seeing an increase in P-Selectin after admission to hospital. Next, we have it compared to that injury severity score that I brought up earlier. And as you can see, there's a possible correlation between the severity of the injury and the P-Selectin levels that we're seeing. And our first group is that 4 through 9, because 4 was actually our least injured, like we had no zeros, which would be no injury. Our next grouping is P-Selectin compared to trauma type, those 3 trauma types that I brought up earlier. And as we can see, there's also this possible correlation between that P-Selectin level that we're seeing in the polytrauma versus our traumatic brain injuries. So at this point, all of the conclusions drawn are preliminary. This is preliminary data just because we have more samples to add and really going to look statistically at the differences between trauma types. However, at initial review, there seems to be some level of platelet activation in trauma patients. That degree of function cannot really be said at this point, but there does seem to be at least some activation. Going on from here, further research should confirm this, and one of the ways that it may be done is going and looking at these activation-based proteins rather than previous studies which have used agitation methods in which platelets are exposed to an activator. And this may not necessarily be as accurate, just taking into account spent platelets that may no longer react to that activator. Another thing to bring up is that transfusions may potentially change the level of platelet function that we see in trauma patients. And so we should definitely put a focus towards that to see whether or not trauma patients, when they receive transfusion, are having significant differences in the functioning of their platelets and their coagulation. My acknowledgement and thanks go out to Sam Underwood, Dr. Martin Schreiber, and Beth Rick, as well as the entirety of the trauma lab at OHSU, as well as the samples from the AT3 research project. Also Portland State and Bill Exeter, as well as the NIH for funding through these grants. Here are my sources, and then any questions.
Great, thank you so much, Evie. That was an awesome presentation. Yeah, let's give Evie a round of virtual applause. Yeah, really interesting work. And this is also just so important, thinking about injury and healing and kind of what those important factors are. Yeah, absolutely. Thank you so much for the work that you've been doing over the summer. But I'd like to open it up first to the audience. If the audience has any questions for Evie about the work she's been doing. I guess one question, oh, I see one here, yeah. Is this going to be used to determine TBI?
So not quite. The whole point was just to look at whether or not there is platelet activation going on. What the reason that we see a lesser level P-Selectin within TBI patients, I can't really speak to. For determining TBI, I would like to shout out to my other scholars who will be presenting Thursday. And one of them, Rizal, will actually be talking about potential ways of looking at TBI. My research isn't quite that specific.
Awesome, that's a really good question. And me being kind of naive in this area, are there other cell types or factors within kind of that whole microenvironment that might be interacting with the platelets?
Yeah, so platelets are part of the first part of the coagulation cascade, which is how your body is going to try and stop bleeding. That's why I said that initial platelet plug. So there are many different factors and proteins that go on within this kind of activation stage because the platelet pathway is very quick, but it's not quite as efficient as the rest of the coagulation cascade. And so P-Selectin was just chosen as it's one of those activation proteins. Whether or not another one could be more accurate or if it's stable for longer, I couldn't really say, but it's definitely just one of the many different things that go on when your body is attempting to stop bleeding.
Awesome, that's really, really interesting. Yeah, I'd love to learn more about this too. Yeah, does anyone else in the audience have any more questions for Evie about this? I guess one last question would be, what is your next step in the project and what are you excited about kind of finding out with your experiments?
Yeah, the next step is sort of, like I said, statistical analysis for these different groups, really looking to see if there is correlation between the severity of injury and that increased P-Selectin that we're seeing. And then after that, it would be to start writing. Essentially, that's my next sort of step after I do that. I'm really excited though, because that sort of coagulopathy where blood isn't coagulating properly, I'm not sure if I explained that clearly, where the body isn't coagulating or isn't coagulating properly or as expected, to be able to say that there is function is really, in my opinion, just cool. To be like, our bodies are doing what we want them to do. It just maybe something else is going on in that time frame.
Awesome. Really great job, Evie. Really, really important and interesting work. I'm excited for the future results.
Thank you.
Let's give Evie another round of virtual applause. That was a great, great presentation. Thank you so much. All right. We have 1 minute for our next speaker. to get ready and get set up, our next speaker is Alicia Arreola-Bustos. Alicia, if you're here and you'd like to get ready and start sharing your screen, you can let me know when you're all ready to go.
Okay, cool. And that looks fine. Let me make sure.
That looks great.
Okay, cool. So hi, my name is Alicia Arreola-Bustos, and I'm part of the Derek Lamb Lab, and my study is called Validation of Sleep Questionnaires in the Down Syndrome Population. So my study primarily works with pediatric Down syndrome population, and we specifically look into obstructive sleep apnea. And the goal of our study is to validate a sleep questionnaire that is currently being used as a screening diagnostic tool for obstructive sleep apnea in pediatric departments. It is called Sleep Disordered Breathing Subscale of the Pediatric Sleep Questionnaire, or also PSQ. The more common screening tool that we use is actually an overnight sleep study. And this questionnaire is just kind of a better tool. It's more accessible and a lot easier than having a child try to sleep through with a bunch of monitors. And we will actually be comparing the two to see if the results align in order to validate it into the Down syndrome population. And the other sleep study that we do, the other questionnaire that we study is also the OSA 18, which is a quality of life questionnaire that specifically pertains to sleep. And that one is compared to our generic pediatric quality of life questionnaire. And those two will be studied as well in order to see if they are validated in the Down syndrome population. So we're predicting that the thresholds in the Down syndrome population are going to be different than those in the non-syndromic population, just because it's a lot, obstructive sleep apnea is a lot more prevalent, and there are some behavioral issues that they may experience that might not necessarily align that are part of the questionnaire. So we don't think that they will necessarily align, but for this quality of life questionnaires, the OSA 18 and the PEATS-QL, we believe that those two will be associated and will align pretty well. So we primarily enroll patients from the Down syndrome clinic here at OHSU, where we see about 3 to 6 patients a week, just on Fridays. It's a multidisciplinary clinic, so we don't see too many patients. But once I've gone through the inclusion and exclusion criteria, I reach out to the parents and I kind of contact them about enrolling into the study. Yeah, we've had a lot of difficulties due to COVID trying to enroll patients, so we haven't had the best of luck. We just recently switched from remote work to in-person. We're already starting to see a little bit more success, but it's definitely going a little bit slow. So once patients have been enrolled into the study, we have them fill out our questionnaires. They fill them out twice, once at the initial enrollment, and then again before the sleep study. That it's typically done within 3 months of enrollment. Yeah, so like I mentioned before, we're basically trying to discriminate or eliminate any discriminatory or biased questions that are in the questionnaire that might not necessarily pertain to the Down syndrome population, and create better gold standards that will better help us diagnose children with Down syndrome with obstructive sleep apnea. Although it's highly prevalent, a lot of children still don't get the proper diagnosis because they just can't go through a sleep study. It's kind of a hassle for them to do that. And because of the comorbidities that obstructive sleep apnea does have, it is really important for us to be able to give them proper diagnosis and proper treatment options as well. So lastly, I'd like to thank Dr. Derek Lamb, my PI, Dr. Joseph Pinter, Dr. Rula Moala, Sarah Forg, and Eleni O'Neill, which have been all a part of this study, and then as well the NIH Build Exito program. Thank you so much. Do you guys have any questions?
Thank you so much, Alicia. That was a great presentation. Let's give Alicia a round of applause. That was really interesting work. Again, really, really important in kind of understanding what's taking place with Down syndrome and around sleep apnea. Yeah, I'd love to open it up to our audience if the audience has any questions for your research. And I guess I can kind of start us off, and this might be me not knowing the field as well, but in your group, I know most of your work is around pediatrics and young ages. It looked like up to around 18. I'm wondering if there's been comparisons made with different age groups in looking at some of the findings that you're discovering.
Yeah, so I'm not aware of any studies that have been done particularly with the sleep studies and the questionnaires. I do know that adults, they do have some studies around just obstructive sleep apnea in general. I think they tolerate a lot more than younger children do and are a lot more willing to work with researchers. I think somebody asked the same question in the chat.
Yeah.
We personally aren't looking at doing adults just yet, but I think just kind of going off of what we are collecting and what our results are, I think that definitely would be something that might be helpful.
Yeah, really interesting. I'm also seeing there's a hand raised. Anaya, did you want to unmute yourself and ask?
Yeah, sure. And I was just wondering, and I don't know a lot, but is there any like intersection between people on the autism spectrum and sleep apnea? And have you come across that in your work? Just because I am on this website where people on the autism spectrum who are children specifically, and there's this like huge issue with sleep, and so I just wanted to see if you had sort of anecdotally like heard of any sort of crossover in the work.
Yeah, so this specific study, we have not done that, but I am a part of a different project with the same lab that we have found some sort of like cognitive deficiencies or just neurological development issues when it comes to sleep apnea in children. So I would say maybe, I can't confirm specifically that autism, but there are definitely, it does affect some sort of neurological development to an extent.
Wow, okay.
Yes.
Cool.
I'm also seeing there is a comment. This is exciting work since sleep apnea is a comorbidity for heart-related diseases. So there's a great correlation there with that. And then another question is, would there be a way for children that cannot take the sleep study in person to conduct it at home?
Oh, that's actually a good question. We haven't figured out a way to do that yet. Our sleep studies actually take place at the Marriott Hotel in Portland. They are allowed to have a parent there, but I do know that it is a challenge for children with Down syndrome to just like change their environment, and that is not something that we have done yet, but definitely something we should look into.
Yeah, really interesting research. Yeah, does anyone else have any other questions for Alicia? That was really, really important work. Yeah, I guess, what are you most excited about and kind of what your next steps are going to be in moving forward?
I think I'm most excited to see if the questionnaire is something that we can use as a diagnostic tool, just because having them undergo the sleep study is really rigorous on them sometimes, and it's really challenging. So if we're able to just use a simple questionnaire to diagnose them, I think we'd get the results a lot faster and just be able to do that. And I think next step, just being able to publish that and hopefully find,
Yeah, great, beautiful research. Thank you so much, Alicia. Let's give Alicia another round of applause for that great presentation. Thank you very much. And we have another minute to get ready for our next speaker. So our next speaker today is Jasmin Mabry. Jasmin, if you're here and you'd like to start sharing your screen, let me know when you're ready to go. Looks good.
All right, you guys can all see it? Okay.
Looks great.
So hello, everyone. My name is Jasmin. I am a psych major with a concentration in neuroscience, and the title of my study is Chronic Cognitive Deficits After Mild Traumatic Brain Injury, which testing methods exhibit the most external validity. My advisors are Lori King, who is my PI, and Cody Campbell from the Balance Disorders Lab in the Neuroscience Department of OHSU. So cognitive deficits are really common following mild traumatic brain injury, but there's limited evidence that standard evaluations are actually useful for detecting impairments, especially in the chronic stages of MTBI. Specifically, the external validity and overall sensitivity of these measurements has been brought into question, especially in chronic MTBI populations where cognitive symptoms are less overt, they're less detectable. So there are two main types of cognitive evaluations at the moment. There's traditional neuropsychological testing methods in which participants undergo seated or stationary tests. These are often conducted on a computer. And there's also functional neuropsychological testing methods in which laboratory or clinical settings kind of attempt to mimic real-world settings by putting participants through more complex testing that engages multiple senses, such as walking while also completing some sort of cognitive task. So this study examined one traditional and one functional cognitive test in order to determine which best discriminates between people with and without chronic MTBI. So the question that I'm asking is, do these tests evaluate what they actually claim to evaluate? So the first cognitive assessment that we examined was the Automated Neuropsychological Assessment Metrics, otherwise known as the ANAM. And this is a seated computerized neuropsychological test, which is aimed at assessing just the general cognitive state and psychomotor performance of individuals suffering from MTBI. So this is the traditional neuropsychological test. It's originally developed by the military, which is why I have this image here. And it's made up of a battery of tests. So it's many different tests kind of one after the other consecutively. That provides us with a composite score from all of the tests combined and tells us whether or not someone has cognitive deficits and the extent of these cognitive deficits. Participants also underwent a motor cognitive dual task test, which is a functional neuropsychological test. So the cognitive aspect of this test was the auditory Stroop, in which the participant listens to a voice saying the words high and low in randomized pitches of high or low. And the participant then has to verbally state the pitch of the voice while ignoring the spoken word. So it's a little confusing. The dual task test combines this Stroop test with a simultaneous, continuous, simple walking task of 13 meters, as you can see in the diagram below. And for these evaluations, we documented gait speed as recorded by inertial sensors that are placed all over the participant's body. And we also documented the accuracy of answers in the Stroop test. So whether or not someone was actually stating the pitch of the voice, or which would be the correct answer, or whether they were accidentally stating what the voice was actually saying, so high or low. And then if one or both of these categories suffered, it is indicative of cognitive deficits. There's a more complex way to calculate this, but that's the easy way to say that. So this study was actually a secondary data analysis in which AUC, or area under curve, from receiver operating characteristic curves were used to identify which outcomes best discriminated between groups. So using this method, we were able to evaluate whether or not each cognitive test was able to actually determine what it claimed to determine. So in this case, that would be the presence of an MTBI. The study was conducted using 51 MTBI participants who were at least 3 months out from injury, which would classify them as chronic MTBI patients. And we also had 58 age and sex-matched control subjects. So our results indicated that dual task gait speed was the best at discriminating between groups with an AUC of 0.75. The ANAM composite scores were second best with an AUC of 0.73, and the dual task Stroop accuracy did not differentiate between groups at all. And so a reminder that with AUC scores, a 1 would be a perfect test. So the closer that these scores are to 1, the better they are at discriminating between groups. So for people with, first and foremost, our results show us that for people with chronic MTBI, cognitive deficits may still be present months after the initial injury. This is a widely unexplored area. And these deficits can be detected using neuropsychological evaluations. So while people with MTBI demonstrate both motor and cognitive deficits during dual task conditions, only motor dual task assessments could differentiate between groups during our functional neuropsychological evaluation. This might be because the Stroop task was too easy, so that would be something that we could explore in the future. And another next step to be taken in this research is to look at the prioritization of motor function over cognitive function that we saw in our functional neuropsychological testing and figure out whether this is a pattern of behavior and what the implications of this behavior are. All right, I would like to thank the OHSU Balanced Disorders Lab and the Build Exito program for providing me with the opportunity to do this research. And I would also like to thank my PI, Lori King, and my lab mentor, Cody Campbell. Here are my references. And if anyone has any questions, you can feel free to ask them.
Great work, Jasmine. Yeah, really interesting presentation. And I love your use of your figures and graphics. Really, really nice. Yeah, let's give Jasmine a round of applause for that awesome work. So yeah, I'd like to open it up to the audience. If the audience has any questions for Jasmine about this research to start off. And I can maybe start us off with one. I guess one question I have, and you may have mentioned this and I might have missed it, is are there plans to perform these tests with the same individuals over time after the traumatic brain injury? So maybe, you know, years later and seeing like how those results change.
Sorry, my internet connection is unstable, so most of that question cut out. Could you ask that again?
Yeah, absolutely. Sorry about that. I was just wondering if there are plans to perform those tests on the same individual over time to see how those results can change after maybe, let's say, a year, 2 years after somebody's experienced a brain injury.
Yeah, so the participants that are furthest out, I believe, are like over 1,000 days post-injury. When it comes to that far out, these tests are mostly unable to detect cognitive deficits if there are any, which we're unsure of. So I believe that the most far out that we test is usually about 6 months out. And then after that, the tests are not good at picking up any long-term cognitive deficits.
Wow, it's really interesting. Yeah, I'm seeing another question in the chat. What kind of cognitive deficits were most common in the study?
So there is like just an overall lack of function when it comes to, first of all, speed of cognition and also impulse control. The kind of stuff that takes kind of a higher level of thinking. So there's a lower level of thinking in which things happen kind of automatically. And then there's a higher level of thinking in which you actually have to cognitively process things. And there were actually deficits in both. So it was just overall cognitive utility was down.
Oh, thank you for that. Yeah, really, really interesting work. Yeah, I'd be excited to see how this actually plays out and can be used kind of more, you know, broad spectrum in the clinic, and how this can, you know, help a big variety of people. So yeah, thank you for the work that you're doing. This is really great. Let's give Jasmine another round of applause for a really great presentation today.
Thank you.
All right, we have our last but not least, our final speaker for the symposium today is Araya Green. Araya, if you're here and you'd like to go ahead and get your presentation set up. Let me know when you're ready. Looks perfect. Yeah, go ahead.
Okay. Hi, my name is Oriah Green, and I'm doing my project, or I did my experiment on cryopreservation, basically looking at vitrification and thawing. So to start off, a follicle is a sac in the ovary that contains one immature egg. When the menstrual cycle occurs, these follicles release the egg in the result of possible fertilization. Ovarian tissue cryopreservation is the only option for preserving fertility in prepubertal and young female cancer patients that require immediate treatment. Cancer treatments can be harmful to the ovaries and to the follicles specifically, and can lead to long-term health risk, including ovarian failure. The objective of this experiment was to compare both stages of cryopreservation, introducing and removing the cryoprotectant, in hopes of seeking what or which of the two steps cause the most significant damage to the ovarian tissue that is being preserved. The overall goal is to improve ovarian tissue cryopreservation protocol. The vitrification in this experiment was done before it got to my lab. Yeah, I just want to include that. So vitrification is another word for freezing. When a sample is vitrified, it turns into an ice-like structure, and when thawed, the ice-like structure is no longer, which is the thawing process. The staining that you'll see in the pictures were used using the hematoxylin and eosin staining method, or H&E. The experimental groups in this experiment are the control, which is no vitrification or no freezing; vitrified thawed, which is the process of introducing the cryoprotectant to the tissue; and vitrified thawed washed is the removal of the cryoprotectant. So on the right side of the presentation, you can see images that were taken using a microscope of the slides that I stained using the H&E method. The first column shows the control group, which is how oocytes normally look when they're not vitrified or thawed. The middle column shows the follicles after being vitrified. So in the vitrified state, as you can see, it resembles the control closely. And if you look at the last column, it shows that the thawing process, is where things start to look different in comparison to the control. As you can see, a lot of the white spaces that are not supposed to be there, and that is a result of the follicle shrinking once the cryopreservation is removed, and that is a sign of osmotic damage. So the next steps for this protocol, or for the protocol that was used during the vitrification method, is to modify the second half, which is the removal of the cryoprotectant, being that it is the main cause of the tissue damage.
Sorry, I couldn't get off of mute. That was a great presentation. Thank you so much, Oriah. Yeah, let's give Oriah a round of applause for that presentation. Really, really interesting work. And I'm really interested in this because you're looking at that freeze thaw and what happens to that tissue. And that's something, you know, being a scientist and being in the lab, kind of thinking about how do you actually preserve tissues in the best way. I first want to open it up to the audience. I don't want to bombard the questions. Does anyone in the audience have a question for Oriah about her presentation and the preservation process? I can start us off. I'm interested in if you could talk a little bit about, like, what are the current methods and how does this research really hope to improve on that? Kind of what might those next steps be that might, you know, make the preservation process even better to help preserve that tissue?
Yeah, so what my lab did, we didn't really focus on the vitrifying process. It was more so as of a secondhand check of the process that was done before reaching my lab. So I wish I had more information on that, on the protocol, but we mostly just did the staining and the imaging that was shown on the slides.
Awesome, awesome. And when you were doing this research, Yeah, I was just interested about also the staining and the tissues. Were you able to do a lot of that staining yourself in the lab? And
Yeah.
were there any challenges there that you faced with that?
So this experiment was conducted by myself. The imaging, the staining. It was kind of difficult in terms of the staining because it's very precise. You don't want to remove the tissue when you're staining, and all of the details that go into that. I think that was the hardest part.
Yeah, really nice work. Yeah, that's a really
Thank you.
difficult process for sure. So great skill there. Um, Yeah, and I'm looking to see. Oh, I see a question in the chat. Were there differences between individual thawed groups?
That's a good question. So what we did, I forgot to include it on the slides, but uh, I wanted to note that we didn't have a large enough sample to actually confirm or deny the protocol in a manner that would be kind of accepted in the public. But for what we did, we used one sample per one sample of an animal, because my work is at the primate center. So we use a species of animal, but to get more information on that, we probably would need to do different species and more animals overall.
Awesome. Really interesting and really great work. These are some really important techniques that you're
Thank you.
learning in your lab. So really nice work. Yeah, and if there aren't any more questions, yeah, is there anything else you're excited about with this research moving forward in the lab?
Yeah, I'm just excited to see what's next, and maybe get some hands-on experience with the vitrification process.
Awesome. Well, thank you so much, Araya. That's a great
Thank you.
presentation. Yeah, everybody, let's give Araya another round of applause for a really beautiful presentation today. That's the last of our presentations for the first day of our 2021 Exito Research Symposium. So thank you all so much for joining us, for coming and showing your support for your cohort and your peers and your lab mates. And thank you so much for the RLC mentors for coming and supporting your scholars. Everyone has worked so hard over this summer, and we are just so proud of everything that you've been able to do. I can't wait to hear more about how the research continues. So thank you all so much for doing such great work. Yeah, and we will have more presentations on Thursday, and we'll keep it going with our multiple rooms, and also look out for all the presentations. They've been recorded and they'll be all posted next week. So if you missed anything, you can go back and you can view all of your peers' presentations. So we'll have all those there, and posters as well. So don't forget to check out the posters. So thank you all so much for coming today. And yeah, you've all done it. So now you've had your presentation. Have a great rest of your time before starting in fall. Yeah, we're really proud of you. So thank you all so much. That concludes today's presentation symposium. And yeah, we look forward to seeing you on Thursday.